Thanks.
I'm very glad to have a chance to talk to you guys. And then I'm Boon Leong Lim. I spent 3 years in Oxford and obtained my PhD from 1991 to 1994, and that was me. And then my PhD supervisor was Professor Ken Reed of the Department of Biochemistry. And then this photo was taken when I first report to his lab, and this was my small bench. And I had a very good time in Oxford. And then in addition to me, there were a few PhD students from Hong Kong. And after they complete their PhD or postdoc in Oxford, they returned to Hong Kong, and now they are professors in different universities of Hong Kong. For example, Professor Thomas Leung, he is a professor in the Hong Kong Polytechnic University. Professor Ken Yong is a professor in the Hong Kong Baptist University. And Professor Dennis Low, he just received a 2021 Breakthrough Prize for his contribution in non-invasive prenatal testing. I still remember that in 1993, he always came to St. Cross College because he was chasing after his wife. And then once I asked him, what are you doing? And he said he was trying to detect and sequence fetal DNA from the mother's plasma. And then later, a few years later, he succeeded. And then our current dean of the Faculty of Science of University of Hong Kong, he also worked at the University of Oxford from 1991 to 1993. So there are a lot of Oxford people in Hong Kong. All right, so coming back to my talk, the title of my talk is Revisiting Photosynthesis by in Planta Energy Sensors. So if we want to measure some metabolites like ATP, in the past, we need to extract the metabolite from tissues and then measure the metabolite by some in vitro methods, such as enzymatic methods, bioluminescence, HPLC, or mass spec. However, these in vitro methods require the extraction of metabolite from tissues before measurement. So these methods cannot determine the levels of these metabolites in different cells, and then these methods cannot determine the levels of these metabolites in different subcellular compartments. And these methods are not real time because we need to do the extraction first and then carry out the measurement. And then they do not allow us to carry out in planta measurement. So there were a lot of limitations for those in vitro measurements. So a solution is to transform some ratiometric metabolite protein sensor into plants. Those sensors allow us to measure or observe instant changes of these metabolites in planta, and we can also observe the changes in different cells in the tissues or in different organelles such as chloroplast, cytosol, mitochondria, or peroxisome. So now there are many different ratiometric fluorescence biosensors, and their characteristics include they are specific to their target metabolites. They have a large dynamic range of changes. They have a quick response rate, high fluorescence signals, reversible binding to the targets, and their excitation and emission wavelength should not overlap with autofluorescence. And we need to also pay attention to the effect of pH, as well as for some sensors, we can have a range of sensors that have different KD. And we need to pick the sensor that have a suitable KD for the physiological analyte concentration. And then nowadays, those ratiometric fluorescence sensors can be classified into two groups based on their mechanism, including the FRET or the non-FRET based ratiometric sensors. So what is FRET? FRET is that for a simple fluorescence protein, when we use a laser to excite it, when the electron jump back to the ground state, it will emit some energy. in terms of light energy. So this will emit fluorescence. However, if we put a donor fluorescence protein and an acceptor fluorescence protein together, if we link them together, then the fluorescence emitted by the donor or the light energy here actually can be used to excite the acceptor so that the acceptor can have another wavelength emission. And this is called Foster resonance energy transfer. And in short, it is called FRET. And then this FRET efficiency is dependent on the distance between the donor and the acceptor. When they are close, the efficiency is high, but if they are far apart, the efficiency will be low. So nowadays, there are many different donor and acceptor pair, and they can be used for measuring the distance or for a dynamic range within 10 nanometer. So let me take the ATP sensor that is used in my lab as an example. This is a FRET-based ATP sensor, and its name is called AT1.03. This was developed by a group in Japan, and they applied this in animal system, and then our group used it in the plant system. So the principle of this FRET sensor is that this is the donor, and this is the acceptor, and they are linked by an epsilon subunit that can bind ATP. So when there is ATP bind to the epsilon subunit, the distance between the donor and acceptor will be closer, so that the efficiency of energy transfer will be higher. So in this case, we can see that the donor can emit at 475, whereas the acceptor can emit at 5 to 27. So we can measure the fluorescence emitted at these two wavelengths, and then we can get a ratio. So this ratio will be increased when the ATP level is high, but when there's low ATP, then the distance will be further, and then so the emission at 475 will be higher. So if we look at the emission at these two wavelengths, we can see that when there are more ATP, the emission at 527 will increase, whereas the emission at 475 will decrease. And the advantage of using those ratiometric sensors is that they are independent of protein expression level. So when we overexpress this sensor protein in, say, in a plant or Arabidopsis, then expression level in different cells or in different compartments would be different. So, however, for those ratiometric sensors, we can just look at the ratio and we don't need to worry about the protein expression level. So for these sensors, we calibrate it and we can see that it has a detection range from 0.2 mm of, from 0.2 millimolar of ATP to about 1.4 millimolar of ATP. And this is the dynamic range. However, if the ATP concentration is higher than 1.4, then the sensor becomes saturated. So for each sensor, there is an effective detection limit. Another sensor that is used in my lab is a pH sensor. This pH sensor is non-FRET based. We can excite these sensors by 488 and 405 and then measure the emission at 520. And then we can obtain a ratio, and you can see that this sensor is called CPYFP, and you can see that the ratio will change upon pH change. So we also introduce these sensors into plants. Nowadays, there are many ratiometric sensors that can measure ATP, NADPH, or NADH, NAD ratio, or pH, or glutathione redox potential, calcium, chloride nitrate, glucose, sucrose, or some plant hormones. And the one in red are the sensors that are being used in my lab. So this is a flat-based sensor to measure a plant hormone, APA. You can see that when it is low ratio, it means high APA. When it has high ratio, that means low APA. And we can see its ratio in the root, in the shoot, or even in gut cell. The advantage of using these flat sensors or ratiometric sensor is that we can target them in different subcellular compartments. For example, this is a calcium sensor. It is targeted to cytosol, nucleus, plasma membrane, and tonoplast. And then we can observe the dynamic change of these sensors ratio, okay, in real time. Okay, for example, here we can look at the nucleus of 3 cells that express the calcium sensor. And then this is the time. And then when ADP was added, about 10 minutes, okay, we can see incineration of calcium, okay, in these 3 nucleus, okay, at different duration or different time point. So you can see that the advantage of using these sensors are that they are cell-specific, organelle-specific, they are real-time, and so it can offer us a lot of advantage in plant measurement. So in our lab, first we use the ATP sensor and pH sensors to study photosynthesis. We introduced the pH ATP sensors in chloroplast and cytosol. We failed to express it in mitochondria. So instead, we express the pH sensors in chloroplast and the mitochondrial matrix. And then we use different light and inhibitors to carry out our studies. Before I present our data, I would like to recall you some of the basics of photosynthesis. Here is the photosystem of plants. And there are a photosystem 2 and a photosystem 1. The OEC complex here, upon elimination, can extract electrons from waters, and the electron will pass through PS1, cytochrome B6F, and then PS1, and then eventually the electron can be used to reduce NADP into NADPH. And so this is called a linear electron flow. At the same time, some of the protons are translocated from the stoma to the thylakoid lumen, and then the water also emit proton. These protons can pass through a ATP synthase from the lumen to the stoma, and this can drive the synthesis of ATP. And so this linear electron flow that involves both photosystem 2 and photosystem 1 can generate both ATP and NADPH. It has been calculated that the ratio generated by the linear electron flow is 1.283. In addition to the linear electron flow, there are also some cyclic electron flows. And then these cyclic electron flows can transport protons from the stoma to the lumen. So the proton can generate ATP or can drive ATP production through the ATP synthase. So the cyclic electron flow that involves the photosystem 1 only generate ATP. As we know that in the Calvin cycle, if we want to fix CO2, it will consume both ATP and NADPH. So the NADPH and ATP generated from the photosystem can be used or consumed by the Calvin cycle to generate sugars. And then the ratio of ATP to NADPH, the demand, by carbon fixation is 1.5. So if we look at the supply of ATP to NADPH ratio by the linear electron flow is 1.283, then it seems that there is not enough ATP. So the question is, how do plant chloroplasts obtain additional ATP? Cyclic electron flow, certainly. But how about ATP input from chloroplast, from cytosol? So in 2015, a article published in Nature, they reported that there are energetic coupling between plastid and mitochondria in diatoms. In here, they show that in the chloroplast of diatom, surplus NADPH will be transferred through the malate OAA shuttle to the mitochondria and converted into NADH. So what is the malate OAA shuttle? This can be explained in this figure. So as I have explained from here, okay, there's not enough ATP, but if you reverse this, you can say that there are surplus of NADPH. So in the malate OAA shuttle, the surplus NADPH can be converted into NADP, and this reaction also converts OAA into malate through a NADPH malate dehydrogenase enzyme in the chloroplast. And then this malate can be exported from the chloroplast to the cytosol. And then in this diagram, you can see that malate can be imported into mitochondria. And then in the mitochondria, there is a NADH-dependent malate dehydrogenase that can regenerate OAA and then generate NADH. So through this malate OAA shuttle, surplus NADPH in the chloroplast can be transferred to the mitochondria in the form of NADH. So in this paper, in the diatom, they suggest that surplus NADPH can be exported from the chloroplast and then into the cytosol and then import into the mitochondria through the malate OAA shuttle. And the NADH can be consumed by the mitochondrial electron transport chain to generate ATP. ATP generated in the mitochondria is exported to the cytosol, which is then imported into the chloroplast. And this can supply extra ATP to the Calvin cycle. So in this Nature paper, they conclude that in diatoms, cytosolic ATP can enter chloroplast to supply extra ATP for the Calvin cycle. But how about in plants? 50 years ago, a German scientist published a paper called adenine nucleotide translocation in spinach chloroplast. The conclusion of this paper is that ATP can move in and move out mature chloroplast freely. And this paper has been cited more than 200 times. And then until 2011, in this review paper, it also says that in non-photosynthetic plastids as well as chloroplast, during the night, rely on the import of energy in the form of ATP via the NTT. NTT is the ATP transporter on the chloroplast envelope. So until recently, it has been believed that ATP can move in mature chloroplast. During daytime, it may supply extra ATP to photosynthesis or for photosynthesis, and during nighttime, it can supply ATP to the chloroplast. So after we introduce the ATP sensor into Arabidopsis, and in 10-day-old seedlings, we look at the ATP level in plastid and cytosol. A red color means high ATP. A blue color means low ATP. Our first observation is that in 10 days old seedlings, the ATP in the cytosol is the highest in the cotyledon, and the level exceeds the detection limit of the ATP sensor. And then in hypocotyl, its level is lower, and in the root cytosol, it is lower. However, in the plastid, the ATP concentrations were very low. So we ask a question: why stromal ATP concentration is lower than cytosolic ATP concentration? Is it due to inefficient importation of ATP, or is ATP consumption in chloroplast tremendous? So we look at the ATP concentration in chloroplast at younger seedlings. We can see that stromal ATP gradually decrease when the chloroplast mature. So this is in the plastid. This is 3 days old seedlings, 4 days old seedlings, and 6-day-old seedlings. So you can see that the ATP level in the plastid actually is decreasing when the seedlings mature. So in 2004, a German group discovered the ATP transporter on chloroplast envelope. And then in Arabidopsis genome, there are 2 genes. One is entity 1, another one is entity 2. By using their promoter to express an enzyme, we can look at their expression, and the blue color means the location where they are expressed. We can see that AT entity 1 expressed in the wing, whereas entity 2 is strongly expressed when the seed germinate. And then we can see that it has a very strong expression in the cotyledon, 2 days old, 3 days old. However, at 6 days old, its expression in the cotyledon disappear, and it is only expressed in the young developing leaves as well as in the root. The entity transporter is an antiporter of ATP and ADP-PI. So say if ATP is to be export, then ADP and PI has to be import, or vice versa. So based on this observation, my hypothesis is that there would be a downregulation of ATP transporter in mature chloroplast in order to restrict the importation of ATP from cytosol. To examine this, we isolate chloroplast that contain the sensor, ATP sensor, from 4 days old, 5 day old, 10 day old, 20 day old seedlings or plants. After we isolate the chloroplast that contain the sensor, we add exogenous ATP. So if the chloroplast allow ATP to get into the chloroplast, the sensor ratio will increase. You can see that the ATP actually could get into 4 days old and 5 day old chloroplast and increase the Fred ratio. However, ATP, exogenous ATP, cannot get into 10 days old or 20 days old chloroplast. So this fits with the observation of the gut study, because when the plant grew older, the leaf will not express the entity transporter, and as a result, ATP cannot be imported into the chloroplast. So our result contradicts with the result of this publication 50 years ago. So why? Then we look at the experimental conditions carefully. In the experiment, these scientists isolate mature spinach chloroplast from the spinach bought in the market. After he isolates the mature chloroplast, he adds some radioactive ATP, and he assumed that the radioactive ATP has been imported into the stoma. Then he washed the chloroplast to remove the radioactive ATP outside, and then he adds some non-radiolabeled ATP. He assumed that the cold or the non-radiolabeled, non-radioactive ATP can get into the chloroplast and then exchange the radioactive ATP from the chloroplast so that later he could count the radioactivity in the supernatant. However, because the NTT is a ADP antiporter, so from his result, we can see that ADP, he did try ADP. If the radioactive ATP indeed was in the stoma, then the addition of ADP should be more efficient than addition of ATP for exchange. But this was not the case. So his data actually showed that there was no ATP translocation, but when he added the radioactive ATP, the radioactive ATP just absorbed on the external surface of the chloroplast. When he washed the chloroplast, the radioactive ATP in the supernatant was removed. However, there was still some radioactive ATP absorbed on the surface. Then when he added the cold ATP elutes the radioactive ATP. And ADP is less effective because it has one less phosphate. So luckily, because he also used ATP, so his data actually showed that there was no ATP translocation, but just absorption. And then this is a in planta study. We eliminate the seedings. This is at the cotyledon. And then this is the ATP level in the stoma. The white bar means elimination. We can see that when we eliminate it, the ATP in the chloroplast increase, but when we turn off the light, the ATP concentration drops very quickly. And then when we turn on the light again, it increases again, but when we turn off the light, it drops very quickly. So that means that chloroplasts consume large amount of ATP in the dark. Then we transiently express the ATP entity transporters in protoplasts. Okay, protoplasts were prepared from the plants that have the ATP sensor in the cytosol, and then we transfect either empty vector or the vector that express the NTT transporter. After overnight incubation, we can see that the cytosolic ATP level in the protoplast transfected with the NTT transporter dropped to a low level. Whereas if we transfect the empty vector, the cytosolic ATP level is still quite high. So this experiment showed that if mature chloroplasts still express high concentration of NTT transporter, then it will consume a large amount of cytosolic ATP at night. And this is not energy efficient. We also stably express the NTT transporter in Arabidopsis. We use empty vector as control. We can see that if we overexpress NTT1 or NTT2, the seedings looks a little bit different, okay? A little bit not that green. And then if we grow these seedings in different daylight period, this is short day. Short day means that 8-hour light and 16-hour darkness per day. And long day means 16-hour light and 8-hour darkness per day. You can see that the transgenic line, they express the NADH transporter. If we grill them in short day, they will be smaller. But when we grow them in long day, they will look like the vector control. So that means that if mature chloroplasts still overexpress the NADH transporter all the time, it is not energy efficient. So the major findings that we obtain by using the ATP sensors include chloroplasts consume large amount of ATP in the dark. Unlike unicellular diatom, cytosolic ATP does not enter mature chloroplasts of Arabidopsis, and mature Arabidopsis chloroplasts downregulate NADH transporter to reduce energy expenditure in the dark. The hypothesis is that for diatoms, they are unicellular. They just need to take care about themselves. So, and they do not need to export sucrose. However, for Arabidopsis, it is multicellular. The mesophyll cells, their function is to carry out photosynthesis and then save the carbon in starch and also produce sucrose so that the sucrose can be exported to the younger part of the plant to support the growth. So this is our findings was published in PNAS in 1998, 2018. So based on our findings, we believe that ATP is imported into pulpastid to fuel chloroplast biogenesis. However, when the chloroplast mature, it will downregulate NADH. And then if mature chloroplasts do not import ATP from the cytosol, so how do they obtain ATP in the dark? So we believe that at night, mature chloroplasts obtain ATP through parasitic glycolysis. So come back to this question. How do plant chloroplasts obtain additional ATP for carbon fixation? Cyclic electron flow, yes. ATP import from chloroplasts, no. And then if we look at this ratio again, it seems that there is insufficient ATP. But if we reverse this ratio, we can see that there is surplus of NADPH during photosynthesis. So plant chloroplasts need to export surplus reducing equivalent during photosynthesis so as to balance the NADPH and ATP ratio. In this experiment, we treat the seedlings with different inhibitors of the mitochondrial electron transport chain. And this is the electron transport chain in mitochondria. And this is the complex 1. It can consume NADH and then pass the electrons. And then at the end, it will drive proton translocation. And that proton translocation can drive ATP synthesis in the mitochondria. And complex 2 also can generate electrons. And then complex 1 and complex 2 can be inhibited by rotenone and TTFA respectively. We can see that red, this is the sensors in the cytosol. And red means high ATP level in the cytosol. And this 3 means that its level exceeds the detection limit of the sensor in the cytosol. We can see that if we treat the plant with rotenone to inhibit complex 1 or treat the plants with TTFA to inhibit complex 2, the ATP level in the cytosol will drop to a moderate level. And if we combine both, if we inhibit both, the ATP level in the cytosol after 1 hour will drop to a very low level. And then if we use oligomycin to inhibit the ATP synthase, there is undetectable ATP in the cytosol. So based on this inhibitor treatment, we show that mitochondrion is the major source of cytosolic ATP in the duct. Then we treat the seedlings, 10 days old seedlings, with rotenone so that the cytosolic ATP drop to a detectable range. So after we treat the seedlings with rotenone, the cytosolic ATP dropped to about 0.9 millimolar. And then if we turn on the light, we can see a rapid increase of cytosolic ATP. And this is light dependent because if we also added DCMU, which inhibits the electron transfer in photosystem, then even if we eliminate the seedlings, there is no increase in the cytosolic ATP. So this proves that elimination of chloroplast stimulates mitochondrial ATP production. And in the lower panel, we also treat the seedling with oligomycins that inhibits ATP production in mitochondria. And if we eliminate it, we can still see a increase in the cytosolic ATP, but this ATP is not coming from mitochondria, but we suggest that it is from the triphosphate export from the chloroplast that undergoes glycolysis in the cytosol and generate some ATP. And using the pH sensor in the mitochondrion, and we can see that if we eliminate it, it will alkaline. The matrix, mitochondrial matrix, will become alkaline. When we turn off the light, we will gradually go back to its original level. We turn on the light again, it will become more alkaline again. And this is inhibited by DCMU. So when we combine this observation, we can see that during elimination, there are some reducing equivalents that are transported into mitochondrion, and that can drive ATP production. So what are the source of this reducing equivalents? There are two possible sources of NADH for mitochondrial ETC during C3 photosynthesis. C3 is C3 plants. And then that includes the Maille-OH shuttle that I mentioned before. Second is that there is a glycine decarboxylation in the mitochondria. And there is a process called photorespiration. Photorespiration is a process that, because the rubisco in the chloroplast is, although it can bind CO2, but it is not highly specific. Sometimes it will also bind oxygen. The reason is because the oxygen in the atmosphere, the concentration is much higher than CO2. So in FH, the ratio of binding CO2 to oxygen by rubisco is 3:1. When oxygen is fixed by rubisco, it will generate one molecule called 2-PGA. And this 2-PGA has to be regenerated into 3-PGA by 8 enzymatic steps that are carried out in chloroplast, peroxisome, and mitochondrion. And in the mitochondrion, there is an enzyme called glycine decarboxylase. This glycine decarboxylase can convert two glycine into one serine molecules, and at the same time, it will generate one NADH molecules. So here are two models. Okay, in model 1, you can see that both GDC and Malle-OAA shuttle can supply NADH to the METC. In this model, the surplus NADPH generated in the chloroplast can be transferred through the Malle-OAA shuttle through malate to mitochondrion so that it can generate NADH. And in model 2, photorespiration only is the major source of NADH to the METC, and its supply is larger than the capacity of METC, so that surplus NADH has to be exported from the mitochondria through the Malle-OAA shuttle. So there are two models, and that was from a paper last year. And you can see that in these two models, the direction of the Malle-OAA shuttles are opposite. This is import into mitochondria. This is export from the mitochondria. So actually, there were a lot of paper saying that malate are being imported into malate from chloroplast is import into mitochondria. So you can see that in this 2015 paper, 2014 paper, malate import, 2016 paper, malate import, 2011 paper, malate import into mitochondria. These four papers also reported that there is malate import into mitochondrion, malate import, and in this 2018 paper, surplus NADP, malate is export from the chloroplast and import into mitochondria. And then in these two papers, you can see that in this 2002 paper, GDC in the mitochondrion can generate NADH that can be used by the METC, but surplus NADH can be converted into malate, and malate is export from mitochondrion to cytosol. And in this paper also, malate is export from mitochondria. So two group, right? Two different saying, whether malate is import or export during photosynthesis. To solve this problem, I adopt two sensors. One is NADPH sensors. Another one is a sensor that can measure the NADH to NAD ratio. Both sensors are developed by Professor Yang Yi of ECUST Shanghai, and he applied these sensors in animal system. And our group used these two sensors and introduced them in Arabidopsis. And both of them are ratiometric sensors. And the ratio that we use is 405 and 488. For the NADPH sensors, there are two versions. One is INAP1, another one is INAP4. INAP1 is a high affinity NADPH sensor, whereas INAP4 is a low affinity NADPH sensor. We can see that if we add different concentration of NADPH and in different pH, the emission excited by 405 is independent of pH. So pH would not affect the emission activated by 405. However, the emission activated by 488 is a problem. We can see that pH has a great effect on its emission. So if we take the ratio, we can see that under different pH, the ratio would be different. So this is no good because when there are changes, there could be changes in NADPH level or change in pH. So in order to solve this problem, we have also introduced a non-responsive control sensor called INAP-C. And then if we use INAP-C to carry out normalization, then we can see that both sensors, okay, this is the normalized ratio between 405 and 488. And then this become not pH dependent. And then for sauna that measure NADH NAD ratio before normalization, is it also affected by pH, but after normalization, is it pH independent? So we introduce these sensors, including the control sensors, in different compartments of Arabidopsis. First, we see whether they are correctly targeted to cytosol, partis, and peroxisome, and they are correctly targeted to the destinations. Then we look at the growth of the plants to see whether they will affect the growth of the plant, and luckily there were no effects. So we can use these plants for studies. So first, because there are two sensors for NADPH that have different affinity, INAP1 and INAP4. So we carry out illumination. We look at the light response of these sensors in different compartments of 10 days old cotyledon. This is in cytosol, this is in plastid, and this is in peroxisome. We can see that in cytosol, when we illuminate, there is a decrease in INAP1 ratio, there is a decrease in NADPH, but this is not detected using INAP4. Our interpretation is that the concentration of NADPH in the cytosol is pretty low. So even though if it further decreases, this is not detectable by INAP4 because INAP4 is a low affinity sensor, whereas only INAP1 is sensitive. And for the INAP sensors in chloroplast, we can see that the INAP4 give a stronger change in the ratio. And in peroxisome, the INAP4 in the peroxisome also give a bigger change when we use the INAP4. So based on this data, we conclude that the NADPH concentrations are higher in stoma and peroxisome than in cytosol, because in cytosol only the high affinity sensors are responsive, whereas in the chloroplast and peroxisome, the low affinity sensors are more responsive. Another observation is that illumination increased NADPH concentration in chloroplast, but decreased NADPH concentration in cytosol and decreased NADPH concentration in peroxisome. And for each of these lines, we pick more than one transformant, and then this transformant have different protein expression level, and then we compare their ratio, and we show that expression level of sensors do not affect sensor ratio. And then using different confocal laser power also would not affect the sensor ratio. And then we also show that in planta, the sensors are responsive. For example, if we look at the NADPH sensor in the chloroplast, without treatment, its ratio is like this. If we add some oxidizing agent, the ratio decrease. If we eliminate it, the ratio increase. So that means that the physiological concentration of NADPH in this compartment fits the KD of this sensor, so that sensor can be used. And then we also show that ascorbic acid and glutathione do not interfere with the sensor ratio, so that those sensors are specific to NADPH or NADH-NAD ratio. So then we carry out elimination experiment using 10 days old seedings. We can see that if we eliminate the seedings, the white bar means elimination. And this is the sensor in the chloroplast that detect NADPH. We can see that this is a higher light intensity, this is lower light intensity. So we can see that both light intensity can increase the NADPH level in the stomach, and then when we turn off the light, it will gradually decrease. And here is the NADH-NAD ratio. And then using these two light intensity, we can see that the NADH-NAD ratio also increase. And this increase could be inhibited by DCMU, so that this is light dependent. Then we use an inhibitor called AAN to inhibit the glycine decarboxylase in mitochondria. We can see that if we add the AAN, the increase in chloroplast NADPH and the increase in chloroplast NADH-NAD ratio disappear. This is no inhibitor, and this is with inhibitor. Under AAN treatment, the increases in stomal NADPH and NADH-NAD ratio disappear. But it still increased the stomal ATP production, and then it still induced alkalization of the mitochondrial matrix. In addition to AAN, we also treat the seedings with sodium hydrogen carbonate, because this hydrogen carbonate can increase the CO2 concentration, so that when we use this, there will be no photorespiration, because the rubisco can easily get CO2. We find that we include this sodium hydrogen carbonate. When we eliminate the chloroplast NADPH and chloroplast NADH-NAD ratio also disappear. So based on both AAN and the sodium hydrogen carbonate treatment, when there is no photorespiration, we could not see stomal increase in NADPH or NADH-NAD plus ratio. So based on this observation, we suggest that model 1 is not true, whereas model 2 is correct. So in model 2, photorespiration supply a large amount of NADH to the METC that allow the METC to generate ATP. But there are surplus of NADH that has to be exported through the Marley-OAA shuttle to the cytosol. So the direction of the Marley-OAA shuttle is export of mitochondria, but not import, is the export of Marley from mitochondria, but not the import of Marley from the cytosol into mitochondria. When AAN is applied, there is no NADH generation from the GDC, then the Marley-OAA shuttle will be very effective in transfer the reducing power in the chloroplast to the mitochondrion, and that's why we do not see an increase in NADPH or NADH-NAD ratio in the chloroplast when there is no photorespiration. Other observations that support this conclusion is that in seaweed plant, rubisco pick up CO2 and O2 at a ratio of 3:1. So that means that a lot of 2-PGA has to be regenerated through photorespiration. And GDC is the most abundant soluble enzymes in the C3 mitochondria, comprising approximately 40% of mitochondrial matrix protein. So why that require a lot of GDC is because the photorespiration has a high flux. And if we compare the volume of chloroplast and mitochondria in a plant cell, it has been calculated that the volume of stomach or spinach mesophyll is about 20 times of the volume of mitochondria. That means that the 2-PGA generate in the 20 times bigger chloroplast has to be handled in the tiny mitochondria. And we can also see that Male actually, when we turn on the light, it will accumulate in the plant cells. So that means that Male are being accumulated during photosynthesis. So based on this observation, we propose that during photosynthesis, because of the ratio of there is surplus of NADPH. So surplus NADPH has to be exported from the chloroplast to regenerate NADP. At the same time, the photorespiration also generates a large amount of NADH in the mitochondria that exceed the capacity of the METC, so that the surplus has to be exported from mitochondria in the form of Male. And as a result, Male are being produced by both chloroplast and mitochondria, and that will accumulate in the plant cells during photosynthesis. And this will be important for the regeneration of NADP and NAD as an electron acceptors in both organelles. How much time I have? Oh, all right. So I think I can stop here because I have already talked about an hour. So so I will skip this. Okay. And then I would like to thank my collaborators as well as my students and postdoc. And then, in fact, Hong Kong U also offer a lot of scholarships for PhD. So I hope that you can consider Hong Kong U in addition to Oxford as your destination of PhD study in the future. So I think I should stop here.
Thank you very much for your talk.